Okay, so we have a few answers. Elizabeth said that BMI1 is a proto-oncogene. CDK46 is a proto-oncogene, question mark? CDK46?
Yeah, it's here.
Okay, and then HMGA2 is a proto-oncogene, correct?
Yeah.
Someone said that they have no confidence in that answer, so I'm not sure what to say.
Okay, so just remember, as I told you, the pink genes are proto-oncogenes and the purple genes are tumor suppressor genes. So the pink genes are going to push cells to go through with the cell cycle and divide, and the purple genes are going to prevent them from going through and dividing. So there's a lot of different arrows you see in this picture. So let's go through what each of them mean. So this green arrow, it means activating or promoting. So if you see a green arrow somewhere, so let's say here in cyclin D, CDK4/6, you see a green arrow pointing towards RB where it has one phosphate attached to it. So this means that CDK is promoting RB to be phosphorylated. And then the next one, this red arrow is the inhibiting arrow. So let's say here you see P16. So it has this red arrow pointing towards cyclin D. So that means P16 is inhibiting cyclin D. And then this black arrow over here, these are showing progression. So these aren't three different molecules. It's the same molecule RB, but it's showing that it goes when when CDK4/6 phosphorylates it, it goes and it has one phosphate now, but it's the same molecule. It's not a different protein. And then the next one again shows now it has two phosphates. So this one is showing progression. It's not showing activation or anything, just progression. So the first RB, no phosphates, then it has one phosphate, and then it has two phosphates. And then this like dashed arrow, this shows acting on. So you see this one here, CDK2, and it's going on here, like right here between RB with one phosphate and RB with two phosphates. So this is showing that this CDK2 is allowing RB to have its second phosphate, or it's actually like phosphorylating it for the second time. And that is important because as you see here, RB with no phosphate, it has this inhibiting arrow, and then RB with one phosphate has this inhibiting arrow, but RB with two phosphates doesn't have the inhibiting arrow. And you see the restriction point is right here. So what this means is that unless RB has two phosphates, it's not going to allow the cells to pass the restriction point. Only when it has two phosphates, it's going to allow the cells to pass the restriction point.
Someone in the chat box asked, what about the PDK? It's a pink indicating it's a proto-oncogene, but it is an inhibited red line. Oh, sorry, P13K.
Yeah, so these genes, they're proto-oncogenes, that's what they're meant to do, but in this specific metabolic cycle, the inhibiting and the promoting, it has like a lot of different factors acting upon it. So see, this one is inhibiting the tumor suppressor because its job is to promote the cell cycle. That's why it's inhibiting the purple gene, which prevents the cells to go through the cell cycle. So it's doing its job, it's promoting the cell cycle. So this is what all these arrows mean. So then let's go about how we would analyze this image or how we would answer a question. So my question is, what if we remove BMI1, which is right here? What if we remove this protein from this cycle? What's gonna happen? Okay, so what if we remove BMI1? So try to see what BMI1 is doing and then see if we don't have BMI1, how is it gonna affect the next protein in that cycle?
We have an answer. Someone said the production of P16 would continue? Question mark?
Yeah, so you're on the right track. So because BMI1 inhibits P19, did you see P19?
P16.
Okay, so BMI1 is inhibiting P19, and HGMA2 is inhibiting P16. So these are going in across. So BMI1 is inhibiting P19. So yeah, if P19, if BMI1 isn't there, then there's nothing to inhibit P19, so you would have more P19. Yeah, so that's exactly how you go through this, but then you see like now we have more P19, so we're going to see how that's going to affect it, the next one, and then we have to go all the way to the end of the cell cycle to see how it affects the cell cycle. So let's make it simple by just drawing out the BMI1 pathway. So BMI1 inhibits P19, and then P19 activates or is acting on P53, and then P53 is activating KIP, and then KIP is inhibiting cyclin E, and cyclin E is phosphorylating RB for the second time, which allows the cells to go through the cell cycle. So what happens is we have BMI1, and we're taking BMI1 out, right? And it inhibits P19. So if we take BMI1 out, there's nothing inhibiting P19, so we're going to have a lot of P19. Now, how's that going to affect the next one? So P19 is activating or acting on P53, right? And we have a lot of P19, and it's acting on P53, so then we would have a lot of P53, right? Question?
Yeah, we have a question. Someone asked that if HMGA2 inhibits only P16.
Yeah, so the two here, they're going in across. So BMI1 is inhibiting P19, and HGMA2 is inhibiting P16. Yeah. Okay, so let's come back to this. So we have a lot of P19, which acts on P53, so then we have a lot of P53. And then P53 activates KIP. So if we have a lot of P53, we're going to have a lot of KIP. And then KIP inhibits cyclin E. So if we have a lot of KIP, then it's going to inhibit cyclin E a lot. So then we would have less cyclin E and CDK2. And then cyclin E and CDK2 phosphorylate RB for the second time, allowing the cell cycle to continue. But since we don't have enough cyclin E or CDK2, they are not able to phosphorylate RB for the second time. So we have less phosphorylation, which means that the cell will not pass the restriction point. So this is how you would analyze if they ask you any type of protein. What if we remove it? What if we add more of it? You should go and follow the diagram and see directly what each thing affects the next one and how it affects the next one. And then keep saying this in your mind. So there's no BMI1. That means there's a lot of P19. If there's a lot of P19, that means there's a lot of P53. So that's how you would go about analyzing this.
So we're gonna move on to evolution. So to begin, I would like everyone to define what evolution is. All right, everyone's saying changes in species over time. Yep.
Can anybody say what exactly the change is or where the change is?
Yeah. So what is changing in the species?
Yeah, what kind of change?
Change in heritable. Yeah, heritable characteristics. So to clarify it, it's the allele frequency that are changing. So alleles are heritable. Okay, so the formal definition for evolution is the change in allele frequency in a population over time. So alleles, they're heritable. So in the when two organisms mate and they produce the next generation, they can pass on those alleles to their offsprings. And there are four mechanisms to evolution. The first one is natural selection. The next one is mutation. Then the other one is gene flow. And last but not least, we have genetic drift. I'll go on to talk about each of the mechanisms in detail. Okay. So let's start out with natural selection. Can you guys think of an example where an example of natural selection? White moths survive more than black moths. Okay, using your example, let's see. I'm going to draw it out. Okay, so we have. Okay, let's say that this red dot is an organism. and the color is red, right? And we also have a different type of organism that has the blue color. Changing back and forth. Right. So there are lots and lots of organisms in a population. So a lot of reds. And there are also a lot of blue. Okay, one day a predator comes in and it only sees the blue organism because red is blended in with the pink background. Yes. So what's going to happen is that all the blue organisms are going to be eaten by the predator, only leaving the red organisms.
Because the red were able to blend in with the
Background.
red background of our PowerPoint.
Mm-hmm. And over time, only because all the blue organisms are gone, only the red organisms are going to continue to reproduce. And in the next generation, you're only going to see the red organisms. That's an example of natural selection. All right. So how does natural selection occur? All right, so one, we have overproduction. Overproduction happens every generation. So each generation, you're going to have a lot of offsprings, and the number of offspring is going to be more than what the environment can support. Next is variation in the population. So in each generation, there are always going to be different types of offspring. So they're going to be a little bit different from each other. That's what makes them unique.
Yeah, so different in their fitness,
Yeah,
not like the way that they look or anything.
And there is competition for resources. Like I said before, overproduction causes shortage in resources. So there's always going to be competition to like survive, to fight for the resources. So like resources such as water, food, anything.
And then who is gonna survive that?
Only the fit.
Yeah, only the fit ones.
Yes.
Out of the overproduction. So when you overproduce, you have a lot of offspring. Not all of them are going to be fit for the environment. So the ones who aren't fit enough, they're going to die. And then the ones who are fit, they're going to win in the competition for resources, and then they're going to go on.
Yep. And number 4 is adaptive advantage. Because of variation, the organisms are going to be different in their, let's say, different in their like fitness. So what's going to happen is that one organism might have a little bit advantage over the other, and only those who are the fittest can survive in that environment. Number 5 is reproductive advantage. Only those who survive the competition can reproduce, and they're gonna keep reproducing. So there's always going to be a change in the population.
Yeah. So if the fit ones reproduce, then they're going to pass on their fit genes, and then their offspring are going to be fit. So that's how over time, you're going to end up with a fit population.
Mm-hmm. For natural selection to occur, you need these 5 tenets to happen.
All of them.
Yeah.
And they all kind of link with each other.
Moving on to mutation. Mutation is one of the 4 mechanisms. So what's going to happen is that in each generation, there's going to be an error in DNA replication. So what's going to happen is that you're going to have like a DNA sequence A, T, C, G. But when DNA polymerase replicates your DNA, there's going to be an error. So it could be another A, T, G, C. And because of this mutation, the organism is going to have a different type of trait, and that trait is going to allow the organism to either survive or die off in the next generation, basically.
So it can be advantageous or it can be disadvantageous. And sometimes mutation doesn't affect anything.
All right, so moving on to the next tenet is gene flow. So an example of gene flow is when you have an organism that is different, an organism that is different from a population. So let's see. So you have this organism that is not part of the original population. And I'm going to color it blue. And then we have a bunch of red species. In gene flow, this blue organism is going to migrate into this population, and the members of this red population is going to mate with this blue organism. So what's going to happen is that in the next generation, you're going to have a different type of offspring rather than just having all red offsprings.
So some of them might be blue because now you've introduced this blue allele into the population. And then you already know how Mendelian genetics works. So if it's depending on if it's dominant or recessive, there's going to be different probabilities, but you are going to see some blue individuals in the next one.
Yeah. So last but not least is genetic drift. Genetic drift is when a random event occurs. So it could be anything ranging from a natural disaster. So like, let's say a storm happened and you have these organisms again, these red organisms. But there are some organisms that are different from the others in the population. So when a natural disaster like a storm occurs, it's going to take out all of these special ones and only leaving the red circles organisms. So when that happens, you're not going to see any more of those special organisms in the next generation because they all die off due to a natural disaster, and only leaving the circle ones.
So now that we don't have any more star ones, they can pass on their star genes. So we're not going to see any star organisms in the next generation. And genetic drift doesn't always have to be a natural disaster, but natural disaster is a good example. It could be a disease that was affecting a population and it like killed majority of the population, and only some people who were strong enough to survive the disease or weren't infected with the disease in the first place, they were able to survive, so then they're going to pass on their genes.
Yeah. Another example is when like you have hunters, like for example, you know like people go on huntings, right? And you see those rare animals like bisons and stuff like that. If they're very low in numbers, and when people hunt those organisms, we're going to have only we're not going to have the bisons anymore because they all die off due to like hunting. So like in the next generation, you're not going to see any more bisons, and that's why we like have nature patrols and regulations and stuff like that, so that we don't lose any more organisms than we have to.
Yeah, so you can't hunt for endangered species, and endangered species just mean when there's low number of them already. So you can hunt for them and kill more of them because then they're going to go extinct. So that's why you can hunt for endangered species. Okay. So we know these things about DNA replication so far, without going into how it happens. So we know that it's semi-conservative. So Mendelssohn and Stahl discovered that DNA replication is semi-conservative. So then how it happens is, your DNA is wound together 2 strands in a double helix with the bases are what connect the 2 strands with their hydrogen bonds. So the DNA helix is connected with the hydrogen bonds between the bases. And then we unwind the DNA into 2 separate strands. And then each strand is used as a template. And then its complementary strand is synthesized. And then they wound up together and form a new pair of DNA. So then you end up with 2 identical pairs of DNA. So there's a lot of enzymes involved in this. So the one that actually breaks the hydrogen bonds and separates the 2 strands is called helicase. So this one separates the double helix into 2 separate strands. So then we can go and replicate each of them and synthesize the complementary strand. So helicase separates the double helix into 2 strands, and then there are some enzymes that hold the strands apart because they're still attracted to each other to make the bonds and then form the double helix again. So then single-stranded binding proteins, they hold the strands apart so they don't form their double helix again. And then primase attaches the primers, and the primer is basically just a bunch of RNA bases. And the reason why we attach RNA bases and not DNA bases is because if we add DNA bases, then we're already contributing to making the strand. So we want the primer to be a signal of where we need DNA replication to start. So that's why we have to put RNA, because if we put DNA, it doesn't tell the enzyme what kind of signal it is to start. So that's why I put RNA. And then later the primers are removed, and then we put the DNA bases there again to complete the strand. And then DNA polymerase, it makes the new strand for both of the strands that we just separated. And the thing about DNA polymerase is that it only works in the 5' to 3' direction. And as we know, one strand of DNA is in the 5' to 3' direction, and the other one is 3' to 5' direction.
So what would you call a DNA strand that is working in 5' to 3' direction?
So because DNA polymerase works in 5' to 3' prime direction, the strand that is in the 5' to 3' direction, it is synthesized more smoothly because it's in the direction that DNA polymerase works in. So that strand is called the leading strand because the synthesis is more smooth and more continuous on that one. And the other one, it's a little slow on the other one because that strand is in the 3' to 5' direction. So that strand is called the lagging strand.
So on a lagging strand, because it's only adding DNA in segments, you're going to see fragments that are called Okazaki fragments.
Yeah. And then our DNA ligase joins those fragments together to make a strand. So now let's see it visually. So this is our DNA strand, DNA double helix. So first we want to separate it. So we have DNA helicase here. It goes ahead and separates it into 2 strands. And then I'm gonna label which one is leading and which one is lagging. So 5' to 3' is leading, and then 3' to 5' is the lagging strand. So then we have our DNA polymerase, which works in the 5' to 3' direction. It works smoothly, gives us a new complementary strand for the 5' to 3'. This synthesis is done. Then we have the lagging strand. So it works in fragments there, as Thaw just mentioned, because this is not the natural direction that it prefers to work in. So then we see DNA being synthesized in small pieces. And then it goes through the whole strand, and now we have a bunch of pieces, so we want to join them together and make a DNA strand. So then we have our Okazaki fragments here, and then we have DNA ligase, which goes and joins all these together and gives us a strand, a full strand. And then these make their double helix, and then we have 2 DNA pairs from our one DNA pairs.
So you have one old and one new strand in each of the DNA strands.
Yeah, so that's how it's semi-conservative. And then at the end of replication, because we added the primers before, so the primers get removed and then they're replaced by more DNA bases by DNA polymerase.